You know, when we talk about fighting cancer, we're always searching for that one magic bullet, right? But what if the real breakthrough isn't a single weapon, but a smart, coordinated, two-pronged attack? Today, we're going to dig into some amazing new research that does exactly that, taking on a really stubborn form of lung cancer. So let's dive in. Okay, so the first thing to get is that lung cancer isn't just one disease. It's not one size fits all. And while we've made these huge leaps with targeted therapies for the more common types, there's this group of patients with rare genetic mutations, where the standard treatments just don't pack the same punch. And that leaves everyone in a really tough spot, searching for new options. And that's exactly where this research comes in. It asks a really smart question. What if, instead of holding out for a single superdrug that can do it all, we could combine two different drugs, each with its own unique superpower, creating a synergy that's way more powerful than either one could ever be on its own? All right, so to really get this, we've got to know what we're up against. We're zeroing in on a specific type of what's called non-small cell lung cancer, or NSCLC for short, that's driven by these uncommon EGFR mutations. So what on earth are those? Well, EGFR is a protein that basically tells cells when to grow and divide. In certain lung cancers, a mutation jams that signal into the on position, and the cells just grow out of control. Now, we have drugs to shut that down, but these uncommon mutations, they're like clever variations that have learned how to dodge the standard drugs. And that's the problem, right? This leaves about 10 to 20% of patients with EGFR mutations kind of high and dry, without truly effective treatment options. So, you've got this really stubborn cancer. How do you fight it? Well, the researchers here came up with a pretty brilliant strategy, combining two very different drugs to work together, like a perfectly coordinated team. Okay, let's meet the team. First up is Lazertinib. Think of this as a stealthy precision tool. It's a small molecule that can sneak inside the cancer cell and shut down that faulty EGFR growth signal right at the source. Then you've got Amivantamab. This one's a totally different beast. It's a big antibody that operates from the outside of the cell. And it's a total multitasker. First, it blocks not one, but two growth pathways. And second, it acts like a giant flashing beacon, flagging the cancer cell so the body's own immune system can come in and destroy it. Now, here's where it gets really cool. This isn't just adding one drug to another, like 1 + 1 = 2. When you put them together, something special happens. The combination creates this incredible synergy, unleashing a powerful, three-pronged attack on those cancer cells. Okay, this image here really lays out the whole battle plan for us. You can see the two drugs targeting the cancer cell, and then you see the three key things that happen as a result. Let's break down exactly what's going on. So here is the three-hit combo. First, this dynamic duo slams the brakes on the cell cycle. The cancer cells just can't make copies of themselves anymore. Second, it flips a switch inside the cell that triggers what's called apoptosis, which is basically a programmed self-destruct sequence. The cell destroys itself from the inside out. And third, like we said, it calls for backup, recruiting the body's own immune cells to join the fight and take out the trash. So that all sounds amazing on paper, right? A three-pronged attack. But the big question is, does it actually work? To find out, the researchers put it to the test in some really advanced lab models that were grown from actual patient tumors. And wow, just look at this graph. It tells the story so clearly. It's tracking tumor size over time. The lines up top show tumors that either kept growing or were barely slowed down by other treatments. But that red line at the bottom, that's our combination. You can see the tumor volume doesn't just level off, it absolutely plummets. The tumors are shrinking fast. So the scientists put a number on this, and it is just mind-blowing. It's called tumor growth inhibition, or TGI. For this combo, the TGI was a staggering 185.6%. Now, to put that in perspective, a TGI of 100% is fantastic. It means you've completely stopped the tumor from getting any bigger. A value over 100% means the tumors aren't just stopped, they are actively being destroyed and are regressing. I mean, that's just a phenomenal result in the lab. And if you're still not convinced, check out this waterfall plot. Basically, every bar is one tumor. If the bar goes down below that middle line, the tumors shrink. Now look at that group on the far right, the one treated with our combination therapy. See how every single bar is pointing down? That means every single tumor, 100% of them, shrank. No exceptions in this model. Okay, so the lab results are incredible. Off the charts, really. But here's the moment of truth, the question that really, really matters. Can this success be repeated in actual people? Let's get to the most important part of the story. This is always the big jump in medical research, right? You can have the best data in the world from the lab, but the true measure of success is what happens in the clinic with real patients. So, did it work? Did this combination deliver for people with these uncommon mutations? All right, let's look at case number 1. This is from a 55-year-old patient who was newly diagnosed. Take a look. On the left, that's the scan before treatment. You can see the tumors the arrows are pointing to. And on the right, that's after just 3 months on this combo therapy. The difference is, well, you can see it for yourself. It's just dramatic. To put a number on it, that's a 45% reduction in the tumor size. But here's what's maybe even more important. This wasn't some temporary fix. This patient went 16 months without the cancer progressing. That is a huge deal. Okay, so we know it works for a newly diagnosed patient. But what about someone who's already been through other treatments where the cancer has proven to be even tougher? That's what we see in case number 2. This is a 67-year-old patient whose cancer had already outsmarted another therapy. And again, look at that same incredible story, a clear, powerful response. For this patient, we're looking at a 38% tumor reduction. And this is so critical because it shows the therapy can work even after other drugs have failed. And again, look at the durability. This person remained progression-free for an amazing 18 months. So, where do we go from here? What does all this research really mean for the future of cancer treatment? Well, it points toward a much smarter, more personalized way of doing things. Well, one of the coolest things to come out of this study was a clue about who this therapy works best for. It looks like patients whose cancer cells have higher levels of those EGFR and MET proteins, the exact things the drugs are targeting, they seem to respond even better. Think about what that means. It could let doctors run a test on a patient's tumor before starting treatment to predict, hey, you're a really good candidate for this. That is the heart of personalized medicine. The authors of the paper really nail it in their conclusion. They say this combination could be a new strategy for these patients, filling what they call an unmet medical need. And that's exactly what this is. It's not just a small step. It's a potential game changer for a group that desperately needed one. And that really leaves us with this big, exciting question to chew on. Maybe the future of cancer care isn't about finding that one single magic bullet after all. Maybe it's about becoming master strategists, about learning how to create these intelligent combinations, perfectly tailored to take down a cancer's specific weak spots. Now that's a future worth fighting for.